In order to isolate and culture avian valvular HH14- endocardial cells or HH25 cushion mesenchymal cells, fertile eggs are incubated for 2 days for stage HH14- or 4.5 days for HH25. The embryos are then isolated and the hearts removed. The next step of the procedure is isolation of the HH14- atrioventricular canals and outflow tracts, or the HH25 atrioventricular canals. The HH14- atrioventricular canals and outflow tracts are cut longitudinally, or the HH25 atrioventricular valve cushions are isolated. The HH14- explants are placed lumen side down onto collagen gels. The HH25 valve cushions are digested with trypsin and the cells seated within collagen gels, and both are cultured. Ultimately, these 3D in vitro models mimic key valvular morphogenic events and are useful for deconstructing the mechanisms of early and late-stage valvulogenesis.
Hi, my name is Gretchen Mahler, and I'm in the Biomedical Engineering Department at Cornell University. And the methods you're going to learn today are going to help you answer key questions in the cardiac developmental biology field, such as what factors can initiate and modulate embryonic valvular EMT.
Hey, my name is Russell Gould. I'm here at Cornell University in the Biomedical Engineering Department, working in the Butcher Lab. Before we get started today, it's important to remember that visualization of these techniques are key, as the isolation steps are difficult to learn. This is due to the evolving geometry of the embryonic chick heart and the small scale that these forceps are used with. So let's get started.
To begin this procedure, prepare sterile Earl's balanced salt solution.